the microphones? Well, good afternoon, everybody. Thank you so much for coming to our session. I'm really excited to introduce our symposium that is entitled Dispersal Ecology Meets Island Evolution. Well, yes, basically a few notes about our symposium, but yeah, just before, I would like to thank a lot the organizers for allowing well, by the way, Alain Vanderpoorten, who is the other organizer of this symposium, and myself, Jairo Patiño, to be able to organize the symposium. So I would like to give a huge thank to Claudine Ampen, Olivier Flores, and Dominique Strasberg for, as I said, allowing us to organize this symposium. So basically, yes, during this conference, it has been really insightful to be attending different kind of symposium dealing with the evolution and the importance of life history traits for the different processes and to understand different patterns from species richness to different levels of evolution on islands. And these figures summarize how the dispersal capabilities, this is a paper published a few years ago, or well, yeah, already some years ago, 2012, I think, by Rosie and colleagues, Rosemary Gillespie and colleagues, and basically summarize how dispersal capabilities can have an impact in terms of colonizing, the potential for colonizing islands that are at different geographic isolation levels. And well, this has implications then at the scale of within archipelagos, right? Yeah, no, it sounds like a little bit better. And well, in this other paper, we found a nice summary of how dispersal can have implications for different study areas, since population ecology to evolutionary biology, exactly, biogeography, and also in terms of global chain, in terms of conservation. Yeah, please go ahead. No problem. Basically, this figure is great for our symposium because summarize many aspects, many mechanisms that we are going to try to address with the 5 talks that are involved. We're going to start with Isabel Sanmartin using a model-based approaches to try to understand speciation in different taxonomic groups, from animals to plants, focusing on the case of Macaronaceae. Then we're going to move to a different spatial and temporal scale with Brent Emerson, who is going to be talking about the impact of the importance of drivers of speciation, as I said, at the local microscale. On the island of Tenerife mainly, I think. Then we jump to a different archipelago. We are going to be with Zachary Carter, who is going to be addressing questions related to the impact of geographic isolation on patterns of species richness in animals in different archipelagos of New Zealand. Then we will jump again to a complete different tropical zone, in this case with Tania D'haijère, who is going to be talking about the speciation processes on orchids in the Guinean islands, in particular in São Tomé. And then we are going to finish with Alain Vanderpoorten, who is going to be presenting a new model where, in a species distribution model context, they have managed to implement dispersal kernels to make those species distribution model in a way more accurate to address questions with the impact of climate change. So, well, with this said, it's my pleasure to present to the first speaker, Isabel Sanmartin, and yeah, thank you.
Thank you, Jairo and Alain, for inviting me to be here. So this slide is misleading because I have 3 other co-authors, Johan Nylander, Javier Fuertes, and Fredrik Ronquist. So when I started my career as a young biographer, working with Fredrik Ronquist in 2004, I was studying a pattern that it was, we're studying the patterns of biography in the Southern Hemisphere. We're trying to find whether those patterns in animals and plants fit the breakup of Gondwana, the paleogeographical scenario. So we were using an old-fashioned method at that time. There were no molecular phylogenies with time-calibrated nodes, or there were few for insects. So we used the reconciliation approach. It was called Tree-Fitter. And we have a large data set for that time. We have 54 animal phylogenies and 19 plant phylogenies, and in all we have around 1,300 species. What we found was what we were expecting. There was a previous paper by Peter Linder and Michael Crisp showing something similar in plants. So we found that the animal pattern fit very well Gondwana vicarians, showing a relationship of the animals species that were present in New Zealand, sister to a clade of sister species in Australia and South America, which fit very well the pattern expected from geology, Gondwana vicarians. And we also find a similar pattern in dispersal. So seemed to indicate that Antarctica was very important for animals. Antarctica at that time, we were thinking in the Eocene or even earlier in the Cretaceous. For plants, the pattern was different. We found much more impact on dispersal patterns. So the plants were showing a pattern that was incongruent with biogeography or vicariance analysis, and showing a relationship between New Zealand and Australia. And we now know that it's done due to the west wind drift, the winds moving eastward, and they move spores and seeds in that direction. So plants seem to show a more clear dispersal-driven pattern. A few years earlier, I was working also in another project with a zoographer called Henrik Enghoff, and he was studying the whole Arctic pattern between Europe, Asia, and North America, dividing North America in 2 slices by the Rocky Mountains. And again, we're using phylogenies with no branch lengths, no time scale, and we found nice patterns really fitting very well ancient paleogeography, even the Euramerica, Asia America, Cretaceous pattern, so going back to the late Cretaceous. And years later, Michael Donoghue and Stephen Smith, they are 2 quite well-known botanists, they repeat our analysis using this method called dispersal vicariance analysis. And what they found was similar in a way, but it was also different, because they found a more clear pattern in plants relating, this is the plants, these are the animals, sorry, and this was relating Eastern Asia, that is Japan and China, with Eastern North America. They are separated now by the Pacific Ocean, by the Atlantic Ocean, and the land in between. And the animals show patterns that were more fitting the vicariance, or the transatlantic pattern, the opening of the Atlantic. So again, we found the same difference. Animals showing a more clear vicariance pattern fitting geology, and plants showing a different pattern that didn't fit geology, and it could be explained maybe by dispersal, in this case, dispersal across the Beringian Bridge. So the question that I had at that time, as a young biographer, is, can we say that there are differences between animal and plants, how they assemble in a landmass? Do they have fundamental differences in the processes, either ecological or evolutionary, that govern their community assembly? And if that was the case, then the animals, they are older faunas there, and they get adapted to the plants as the plants are being turned over and renovated over time. That's a very too bold hypothesis. So then I moved into island biography because of different studies, and I found here a really nice scenario to test those hypotheses. And the reason, as we all know, I've been hearing, is that islands, they mostly are dispersal patterns, not vicariance. Mostly for oceanic islands, vicariance is not a pattern that you expect to find between islands. Within islands, there can be vicariance. And then there are oceanic islands, we know, they have been isolated from the mainland. They are a nice laboratory for evolution because of the small size, mostly because we can actually inventory or study the entire biota in a way. And also they have a replicated nature. You have several islands within the island chain. So when I started this, the island biography, of course, the main work was MacArthur and Wilson, island biography theory, and it was groundbreaking, or in founding the quantitative approach to island biography, and we all know this slide showing the interdependence between geographic distance, area, and dispersal rates and extinction rates. As I was working on that, of course, there have been many more works extending the island biography theory. The more prominent is including in Speciation, Lawrence Heney, and the island ontogeny, general island model that has been developed by Robert Whittaker, more recently James Rosindell, including abundance, and we have been here in Rampaletti and Luis Valente with a new method to estimate by maximum livelihood, colonization, immigration, speciation, and extinction. But what I found, what I was looking at that time, is that those models didn't actually recover or reconstruct the island pattern, the within archipelago pattern. I was more interested in how is the assembly of this community by dispersal between islands. So I was more interested on the island archipelago structure, the within archipelago structure. So we use, because at that time molecular phylogenies were available, so we have branch lengths with time-calibrated events. We use what is called stochastic biographic models. They are well known in the literature of morphological evolution. They are called continuous time Markov chain models. Basically, they are memoryless stochastic models in which we have the states. In this case, you could think these are islands, and we have the rate of moving between the states. That could be the dispersal between the islands. And these are governed by this simple Q matrix or instantaneous rate matrix. There is given the rate of moving or between those states. And if you integrate that over the branch lengths of the phylogeny, measuring time, then you get the probability of moving between the islands or between your states. So there have been other models that have been just founded on this basic framework, like the dispersal extinction cladogenesis model of Riri. The problem with those is that very complex, with many areas, I say they just explode. There is no analytical solution for that. So we need to find a simpler model. So we move into a more Bayesian approach in which instead of using a given phylogeny, we co-estimate the phylogeny together with the biography history, the branch lengths, the divergent times, and the states. And we use MCMC simulations to approximate the posterior of the biography history that integrates both the molecular parameters, biographic parameters, branch lengths, as I say, and topology. So what is the core of that model? It's basically a borrowing from the molecular models. So this is the well-known molecular general time reversible model in which you have here the states would be the nucleotides, and the rates are the rates of moving between states or substitution rates. And we have these two parameters in the model when they are time reversible that are stationary and homogeneous. Stationary state frequency, that's the equilibrium frequency of each state, and the relative substitution rate, that's the rate of moving between states. And if we do a simple change, then we find our island model. So instead of nucleotides, we find the biographic states. These are our islands, A, B, C, D, and we want to estimate the rate, probability of moving between the islands. And it's in our molecular models, you can also disentangle these parameters in the Q matrix into two components. One of them is the relative carrying capacity. That would be the number of lineages that could end up in the island equilibrium condition. So you extend this process infinite time, how many species of lineages there would be in that particular island. There is no disturbance, no extinction. And the other parameter is very easy to interpret, is the relative dispersal rate between two islands, for example, I and J. And as all molecular models, they are very flexible. So you can change the models, make them more simpler. They could be like a use Cantor model, equal carrying capacities and rates. There could be one with different carrying capacities, and this could be one in which there are different carrying capacities and different rates. And I just say that 1 year later, the BIS developers, Drummond, Mark Suchard, they developed their own BIB model. They didn't know we exist. So we told them that it was actually already developed. So they cited in the paper, but they did that for tracing viral evolution. But it's basically exactly the same method, the same model. The only thing is that they have another different extensions to use to simplify the model. So, but our interest, as I said, was not on modeling the history of one clade. I wanted to model the history of the biota, sort of the northern hemisphere example, the southern hemisphere example. So how can we use that model to try to find community assembly in a whole biota? So we use the model and we use the implementation in a framework called RefBase, that is a graphical framework for developing methodological or evolutionary models. So what we did is that instead of having one single lineage, we have multiple clades or groups, in this case one bird, one insect, one plant. And because we know that they are very different biologically, we allow them to differ, both in age, in biological traits. For example, some are more dispersed, some are poor dispersed. And also maybe they have different clock rates. They are older or younger, even clock rates within the phylogeny could differ. So we created a hierarchical Bayesian approach in which we try to infer the parameters of my biogeographic model, which would be the covering capacities and dispersal rates, but we allow this to be inferred independently, and then we hierarchize 3 priors to estimate this. So we have very few data for this part, it was only 1 character, but we have all this data in this part, and this hierarchical Bayesian approach allows to have this type of sparse models. And the important part is we allow the dispersal rate to change across clades. We did that in the Canary Islands, and I divide the Canary Islands into 3 different groups of islands according to age. And the interest here, as I said, was to test hypothesis on community assembly. So inspired by a paper by, I think it was Rosemary Gillespie and George Roderick, I developed this type of models I could find reading literature, and it was inspired by other botanists, of course, principally Jair Francisco Ortega. So it has been said that the animals, for example, this is Galotia, they show this pattern in which they move into an island, they speciate another island, they speciate another island, so at the end you have a group of species that are present in different islands. That's in Galotia, for example. In insects, it's more common that you have, once they get to the island, they diversify within the island. So the species in an island, they are sister, and they are sister to a clade in another island. So they have this pattern of within island speciation. Sometimes there is changes like independent colonization, but in plants it was fundamentally different. What they show is that they move between islands, but at the same habitat. So a species in La Gomera would be Laurisilva, sister to the Laurisilva de Tenerife, and Laurisilva de La Palma, and El Hierro. It would be in different islands but same habitat, inter-island colonization of similar habitats. And that very well fits niche conservatism, which is an important pattern in plant community assembly. So together with master students and colleagues at Botanical Garden, we gathered a lot of phylogenies. Some of them were produced by us, and others were just from Gene Bank. And this is just showing that they belong to different families. They were clades that contain at least 3 species. So this was a cladogenesis pattern, a radiation within the archipelago. It couldn't be only 1 species. There was more than 3 or more species. And we did the same with animals. And animals include everything, birds, insects, spiders. And so this is the sample data set. It was very heterogeneous. We have multiple loci per group, and the loci were modeled differently with their protein coding, chloroplast, mitochondria. We needed to account for that heterogeneity to not oversimplify. And we only consider species level or subspecies level if they were in different islands. So it was not population level analysis. And we experiment with different priors to see the influence of the priors. And this is an example of how it's built in RefBase. So it's a model that is feeding from the molecular data set for each individual group, which is modeled independently, but then the biographic part, which is in green here, it will feed from that part. It will borrow strength from that part through hyperpriors, because this is the least informed part of my model. I only have 1 character, just where they are present, the species. I don't have a string of molecular sites, as in the molecular part. So the branch lengths are estimated through the molecular part, but then they are scaled by the dispersal scaler, and then there are a number of dispersal events per site per million year. And this is the model. So it's like hover parameterized lots of. The good thing is that apparently, even though some parameters didn't behave very well, the biographical models, because they borrow from all different groups, they actually behave quite well. So they show good convergence, good mixing, diagnostics, and everything. And what they found is what they expect. So the animals will show large carrying capacities, of course, more for the mainland, and they will show dispersal rates that seem to fit well stepping stone, so there was some exchange between Western and Eastern, but in general it was very stepping stone, and there was almost no dispersal between the Western Islands, for example, and the mainland, which are the farther away, and the same between the central and the mainland. But the most important was the large carrying capacities, and this is the migration rate. So if we just compare to the next one, which is the plants, the first thing we see is that carrying capacities are much smaller. And the migration rates, they are slightly larger and they show a similar pattern. But the carrying capacities are smaller and the migration rate is higher in absolute terms and compared to the animal. So what does it mean? If the carrying capacity is the equilibrium frequencies of the model, they are like rate of not moving away. So it's like equivalent to the number of events when there is no change in geographical area. They can be think as an analogy of speciation within each island. So the more carrying capacity, the more speciation within the island with no changing island. So we found that what we expect to find is that animals show more within island speciation and slightly less dispersal rate, and the plants show the opposite pattern. But I wanted to go further with the idea of niche, of different patterns of community assembly. So the idea is, do plants and animals show fundamentally different patterns? And the most important thing was, do plants actually move more frequently between islands and between habitats? So what I did, I just stratified the islands into the different biomes, and then I estimate the proportion of migration that is explained by island hopping versus habitat switching. And what I found is exactly that. So it's clearly that it is the posterior total rate, and there is much more explained by island hopping than by niche shifting, which means that they fit the idea of, yes, plants, they easily move between islands, but not between habitats. That's much more complex, which fits the ideas of Javier Francisco Ortega in the early 90s. And we also partition by area and geographic distance to see the influence on that. And we did that with the carrying capacities and the dispersal rates. And we found not that so clear results, but it seemed that area was more important for animals than for plants, or the ecological part of the area, and in plants, geographic distance played no role. We thought about the idea of dynamic static models. Our models are all static. So can we change the rate of migration? Because it doesn't make sense that we think in an island archipelago of 20 million years hasn't changed, it's static. All Markov chain models are homogeneous, stationary, time reversible, so they are static, like a framework where you play your dispersal game. It didn't make much sense, because we know, this is a very old slide, but we know from the history of the Canary Islands that it's a hot spot in the mantle, a slow-moving plate, irregular volcanic activity, lots of things going on. So one thing we wanted to test is, can we apply these so-called time heterogeneous models in which you allow the dispersal rate to vary over time? In this case, we ask, when the Pleistocene sea level changed, when it went down, we know that some islands became connected, like Fuerteventura, Lanzarote. Area size increased and distance decreased. Does it have an influence on, can you see that in my phylogeny? So we did that using this type of time heterogeneous models in which we allow the dispersal rate to change between epochs. So we fix epochs in the sense of the different stages in the Canary Island formation, 20 million years, 50 million years, 3.5 million years, and then I allow the last part to change according to a rate multiplier, and I ask, is the rate multiplier positive or negative? Is increase or decrease? That will tell me whether actually dispersal rate increase or decrease in that time period that is the Pleistocene. And I found that, as I say here, it increased much more in plants. Even though plants are not very influenced by geographic distance, in this case, plants show a larger increase compared to the other epochs. And the last question I wanted to ask was, can I also test extinction? So my model has no speciation component, as the models have been shown before. So can I use the carrying capacities to test extinction? And my definition of extinction is different to the classical one in bird death modeling. So we know that the Canary Islands have undergone extinction by in-island volcanism, like Tenerife went through a volcanic eruption 3.5 million years ago, destroyed part of the island, and it was reformed. So as I said before, extinction has been modeled through birth death modeling. This is an example of mass extinction modeling. In this case is the classical approach with a single pulse event. Recently we developed a new model with a time slice approach just to fit better the paleontological meaning of extinction. Can we see extinction within a time interval? But in geography that's very different. Extinction is called extirpation. It means the lineage doesn't go away, it doesn't get extinct, it just gets extinct in a particular island, in a part of the geographic range. So for that, we needed to play with other type of models that have been rarely used in molecular phylogenetics and never in morphological evolution. They are called non-stationary models. So our models, as I said, they are very simple, the ones we use. They are homogeneous, the same dispersal rates over time. They're stationary, which means they reach the equilibrium and then stay there, like here, the burning phase, and they are time reversible. They can be time irreversible, like this case, when you use a directional model. So I wanted to relax that. Particularly, I wanted to relax that part. Can I change the stationary frequency, the equilibrium frequencies of the model? And why is that important? Because if I say the carrying capacities are the in situ diversification, what happens if an island goes through eruption and then loses 90% of the biota? You go to ground zero, you start over again. So it means that suddenly, when we are in equilibrium, we go back to the burning phase. We need to go again and reach the stationary part of the model. In that case, can I allow the carrying capacity to vary over time? And I didn't do it continuously. I did it by stratifying not stratifying the phylogeny, but allowing to change at different breakpoints. So that would be an example of, I can also model like islands that are popping out from the ocean, the other way around, starting with no island, one island. That's also a non-stationary model. And to gain power, I link the animals and plants. So in this case, animals and plants were put together by allowing them to have different migration rate, but they were linked through a log normal hyperprior to gain power. And you will see why. So what did I get? So what I got is that the model seems to be mathematically right, but I have not enough power. So I can see, for example, that at time one, there is you can see here, the estimate that shows that it's very poor. So the mainland would be here, the eastern, time one, there is eastern appears. The second period, the eastern is here, mainland, that's very weird. And then the central is here, and the last time period, current time period, central, mainland eastern, and then this is the western, which is still very young and growing. So they could be showing more like appearance or getting community assembly on the different islands. But the main goal would be, can we estimate the time breaks? Or within those periods, can estimate a point when there is an event of mass extinction? And that's still under development. You can see here some of those posteriors. This is not right. This is completely wrong. So this is something that needs clearly more data, and maybe we work with more other empiricists to get more data, but I think it's still an exciting approach. So to conclude, I will say that stochastic biographic models, these very simple biographic models that really borrow from the molecular phylogenetics, they can be useful for testing hypothesis-driven theories and using phylogenetic data and team data. Whether they can be applied to continents, we don't know. We'll need much more data, of course, and it's more difficult to define boundaries. And then we need more work to test extinction patterns, but I think it could be one of the frontiers to challenge. And with that, I'd like to acknowledge my colleagues, collaborators, mainly Fredrik Ronquist, Javier Fuertes, and Johan Nylander, and the master students, and then the funding Spanish Ministry of Science and the Swedish Science Research Council, and the REP-based developers, Michael Landis and Sebastian Hena. Thank you.
Thank you, Isabel, for this great talk. We have plenty of time for questions, so excuse me.
Well, thanks so much, Isabel, for this amazing and insightful talk. I have just one question for you. I mean, where you present the time heterogeneous BEEB model, and of course it has many more parameters than the standard model. And then my question was, don't we need to first determine whether the more complex model has a better fit to the data than the simpler model before looking at the parameters themselves?
Right. So the simpler model could be the ones I show comparing animals and plants, those seem to have a good behavior. The worst part is to estimate the clock rate. So that's more tricky because we allow different clock rates depending on the group. So I didn't mention the prior sensitivity, but the best prior sensitivity comes when you have a non-independent model. Each group they don't share parameters, but they have a hierarchical model in the sense that all the plants have a hyperprior, so they are constrained by that, animals here, but each plant have their own prior. So that's the model that best works. The time heterogeneous model, the one with the rate multiplier, yes, there is only one more parameter. Okay. That's true. I mean, in a way, we are also stratifying the phylogeny, so we are also introducing constraints. I didn't show anything about base factors because we didn't actually compare that. It should be done. Yeah, that's true. That's a good point. And the last model, yeah, clearly we are far from it, but I thought it was a nice analytical approach.
Thanks very much indeed.
More questions?
Yeah, I'm Ferranseo from Gothenburg University. I was wondering, because in the last part you said you would need more data for the model, and I was wondering how difficult it would be to take this same model and apply it to other archipelagos that already have more data.
Yes, that's a good question. We try to do something, for the first basic model, what I call the simple model, we try to do the same in Hawaii with the data set I have, which is probably not that was something, but I guess Hawaii would work better. And then it comes the question of including not only cladogenetic radiations, but including maybe single archipelago species, allowing for population level data, but that comes with the cost of modeling population level processes. Yeah, I'm not sure whether data will solve it, but I think from the signals, because the best estimate came from the current, which we have more data because we are present time, and as we go back in time, was more difficult to model.
Thank you.
Any other question?
Thanks, Isabel, for such an inspiring talk, as always. So I just have a methodological question. You mentioned that animals speciate more within islands than between islands as compared to plants. Could that be an artifact deriving from the higher polymorphism of the sequence markers applied to animals and the less thorough sampling applied to phylogenies of plants so far? So the sampling is clearly an important, because in animals, so I said we use species level phylogenies, but in animals has been in insects, and the lizards, the Galotia, but it has been a really good level of sampling, so they tend to define varieties, and even sometimes they could even use, whether they were present, for example, in 2 islands, and they were really structured, they could consider them as subspecies, and in plants, if there is not such a phylogenetic in-depth work, then we will miss that. Regarding the markers and the molecular rate, so mitochondrial is faster than chloroplast, and I don't know, because we try to account for that by using different molecular clocks, molecular evolutionary rates. And in plants, we use, of course, ITS, which is faster than chloroplast, and also there were other nuclear markers that were low copy nuclear genes. And when I look at the molecular parameters of each single group, they seem to behave well. The worst part was the clock rate, because we have virtually no fossils, so it's all secondary calibration approach, even molecular rates, and so yeah, that part. Maybe in Hawaii that's will improve. Thank you.
No questions? Well, I have one question, and it's kind of linked to the one that just Julie did. I guess that you have a very heterogeneous groups within all this meta data set, and I don't know, within animals or within plants, depending on the life history traits, did you do like a subset of groups and check if you could get different results?
No, I did not, and I know that was not. Yeah, clearly, my animals were spiders and birds, more sedentary birds, and then in plants I didn't consider they were fleshy fruits or anemochoros or anything, but it's true that you could see from the migration rate, this parameter we could estimate that indicates the dispersal rate, the number of dispersal events per site per unit of time, that it showed that some groups, and so you try to correct that, of course, because you have very short branch lengths with a change in geographic, that there is more certainty in that change that you have very long branch length.
Thank you. Well, I think with this we can thank Isabel again. And now our next speaker is Brent Emerson. He comes from the Spanish Research Council based on Tenerife in the Canary Islands.
Okay, thank you for the invitation to present in this symposium. So today what I'm going to focus on is speciation. It's a topic that our group is interested in, has been for some time. And we know a lot about speciation, but mostly we know a lot about speciation, particularly within arthropods, as a pattern. And we saw that from Isabel's presentation. We know that arthropods tend to speciate a lot within islands compared to plants. Why is that, is the question that we're particularly interested in addressing. And in my abstract, I mentioned I'd talk about two different approaches, but because I overestimated how much I could say in 20 minutes, I'm just going to focus on this community level approach we're taking to address understanding speciation. So this community level approach is just a general sampling framework that we use that allows us to address a bunch of different questions we're interested in, and today I'm just going to focus on the speciation side of that. Okay, my two principal co-authors here are Antonio Salces Castellano and Jairo, and there are a bunch of others involved who I'll mention at the end. So, just going back to that first slide. So, I'm going to present work from the laurel forest of Tenerife, from the arthropod communities in that forest. It's a very small sampling area, but it's very complicated if you want to take a community level approach. It's very hard logistically just to identify all the species there because we're not taxonomic experts on many of the groups. So to deal with this, we developed this protocol. I'm not going to go into detail about it. It's an extension of a protocol developed by Pablo Borges and his lab that has a molecular component. It basically it reduces the taxonomic load and enables us to assign individuals with more confidence to species. But additionally, it also provides us with information about species-specific dispersal limitation. And that's kind of where this talk fits into the symposium, I think. And what do I mean by that? Well, here we have our sampling sites, 10 plots in Tenerife, and this is just some hypothetical examples. Suppose we've got two species that are sampled in all plots, and we've sequenced a mitochondrial gene for a bunch of individuals from each species within each plot. And we've got 5 genetic variants in each. Well, this kind of information could be distributed evenly across all sampling sites. That would tell us that this species approximates a panmictic species. There is very little limiting movement among sites for this species. In fact, there is nothing. In fact, this is an exaggeration of what you would really see. But it's good for this purpose. On the other extreme, we might find that those genetic variants are very geographically structured. And in this extreme example, it would tell us that there are limitations to dispersal among sampling sites. The next question is, why is that? But this is the first departure point for us for understanding something about dispersal history within the species. So we sampled 10 plots across the laurel forest of the Naga Peninsula. You can see it's a crystal cloud forest. You can see the sort of darker, where's the pointer here? See the sort of darker stains along the crest here, that's the laurel forest. And the distance we're talking about here, that's about 14 kilometres from there to there. So it's quite a small spatial scale, really. Now we used, I think Paulo mentioned the sampling approaches in his talk. I'm not going to go into detail. We have replicated sampling across all sites designed to capture as much species richness within the sampling area as we feasibly can. And our focal groups are beetles and spiders, but today I'm just going to talk to you about beetles. Okay, so from our sampling we obtained just over 23,000 specimens. So with our protocol that I just briefly mentioned, we identified and obtained genetic data for 154 presumed biological species. There was a smaller number of species that never yielded sequences, but this is the vast bulk of the community. Just out of interest, 146 of those are native or endemic, so we have a very intact community at the level of beetles. 41 were quite rare, too few samples to do any further molecular analysis, but 105 had enough individuals to allow us to come up and generate some interesting indices of population genetic structure. So here I'm just summarising. We have these two here, which are very not terribly informative, but useful descriptive indices. And this particular approach, which gives us reliable population structure estimates that tell us that the population structure occurred over an evolutionary timescale, not a microevolutionary, but something that involves mutational separation among populations. Okay, now so what I'm summarising here is the results of that last method I mentioned, the BPEC method. And what we're seeing here is like a heat map, a matrix where dark red represents sharing of evolutionary lineages within species among sites. So we can see that it broadly clusters that these four here, very, very similar, whereas these six here are very similar. There's a lot of distinction between them. And we see that summarized here in this hierarchical cluster analysis. And using a permanent we see a lot of the variation that we see there is explained by these two groupings. The groupings we're seeing are basically division between east and west, if you like, separated by a distance of less than 4 kilometers. So how does this look in real data, this little summary? Well, I'm just going to show you some examples that really show it quite well. So the species Lodes oceanica, you can see there's two molecular groups within the species, same molecular groups divided between these two areas in this species, Laparosus subnudosus, Cardiophorus, and I could go on because there is a large number of species that show this, but it gets a bit tedious after a while. We see the same pattern when we look at the data using other techniques for looking at genetic structure, but not so strikingly. It's essentially this first one, this evolutionary one, that really stands out. So then we have this community level structure, if you like, and the next step was to understand what might be driving that. So in this map above is just summarizing the approach we took. We, together with Derek Karger, and Haidor talked about this in his presentation, we generated environmental maps at a scale of 90 meters squared for the variables you can see mentioned here. So elevation, mean annual temperature, mean annual precipitation. So here we see the results for precipitation. You can already see something interesting, that there's quite a striking break in precipitation levels. So we then use landscape resistance analyses. When I say we, I should say Haidor. And what that revealed to us is that for this particular group, the phylo group structuring, we see that the two models that best explain the data above all others are those that include elevation or precipitation. Elevation and precipitation variation explains the structure at the community level that we see. Okay, in the other two indices, it doesn't come out as clearly, but it's very clear with this phylogeographic approach. So our next step was to try and understand at the species level what's going on. So we're just working here with like a crude community level summary, generates hypotheses for us. We want to now test those hypotheses at the individual level. So we generated genomic data for 8 species. We chose 8 species that really showed this pattern quite strikingly with mitochondrial data, and 8 that showed no structure across this regional pattern that we see. Now, here are the 8 species that show this pattern we see here. This is the mitochondrial data that we were working with for the big summary graphics we saw. And in all 8 of them, there's like this clear, striking structure across that region that generates basically 2 mitochondrial clades, 1 in each. Here's the nuclear genomic data for each of those species, and we see exactly the same pattern in each one of them. There's a clear structuring of nuclear genomic variation across this landscape feature. When we look at this same data using principal components analysis, we again see this striking separation of these 2 populations within species. And here we have the output of analysis. I'm not going to go into details about how it works, but it's basically an analysis that lets you assign ancestry to individuals. So in this case, we see ancestry across individuals within each species is assigned to 2 ancestral populations, and these ancestral populations are associated with the east and west, completely consistent with the results we've seen so far. But strikingly and very interesting, I should mention that in one case, we have 3 ancestral populations. In all cases, we find evidence for genomic admixture at geographically intermediate locations. So this is where it starts to become really super non-random. And in all cases, all of these species, their geographic patterns of genomic relatedness at the nuclear level are best explained by topoclimatic variation. Okay. So now we're looking at the 8 species that have no evidence for structure based on mitochondrial data. When we look at the nuclear genomic data, we find that 5 of these are actually structured at the nuclear level across this into these 2 regional populations. Okay, so just ignore these other ones for now. Let's focus on these 5. When we look at them with a principal components analysis, again, we find clear separation between the 2. And even when we look at this ancestral analysis, we find the same thing, that we find evidence for 2 ancestral gene pools associated with the east and the west. And again, signatures for genomic admixture between these 2 that are concentrated in central localities of that peninsula. Okay, so bear in mind again that we're talking a total distance of 14 kilometers here. And again, in each of these 5 cases, it's explained by environmental variation. Okay. So we have a bunch of species that show this pattern, east and west with mixing in these central localities. Another species where it's virtually the same, but the mixing is slightly to the east. And another species where we have a more complex scenario where there's 3 ancestral gene pools that are also mixing at intermediate localities. So this yells out that there's a common driver structuring genetic variation across a large number of the beetle species inhabiting this wet, humid forest ecosystem. Now looking at the temporal time frame for that, just down in this yellow column here, we've got time estimates for when the initiation of divergence within each species started, and it ranges from, I'm pretty bad for this, ranges from back as far as 4.6 million years ago, and that sounds, we had to look at this multiple times to really believe that, but that data, we put a lot of credibility on that, and back through to 2.5 million years, right down to less than 100,000 years ago. So we have a repeated pattern, but the origin of it seems to be not contemporaneous exactly. Where are we going now? So to really understand what might be happening there, or what is happening there, we really have to look at the geological history of the Anaga Peninsula. Now we know that 4.5 million years ago, more or less, there was a large catastrophic mega landslide that removed 15 square kilometers of terrain, which would have rapidly disappeared into the ocean. And that left this interesting geographic feature, this saddle that we see marked here, it would have been persistent through time since that event. If anything, it may become more exaggerated towards the present. So it presents itself as an elevational discontinuity across this dorsal ridge. Okay. Now, what we know about the climate of the Canaries is that this laurel forest, especially this wet, humid component of it, is very reliant on cloud cover during summer months. Cloud cover from what's called in Spanish the mar de nubes, which is a sea of clouds, and I've just got a little animation that I stole from the internet to show you here. Let's just see. So you can see it varies annually in terms of its altitude, and it even varies daily, and so we can just see how much it moves here over the course of a day in terms of its altitude. Now also, we know that it must vary over glacial timescales as well. We just don't really know much about how it varies, but as we pass from glacial to interglacial to glacial to interglacial, the lower limits of that cloud level will have changed as a function of the changing temperatures and climatic conditions that occurred as we pass from glacials in and out of glacial cycles. Now, to really understand in finer detail what that dynamic might be, we've sampled in more detail this species. Just to make sure I don't 35, 45, 50, I think I've got a bit of time. We sampled in more detail this species here, Cales globuli penis. So it's I should have mentioned all these species are flightless. Okay, so we sampled the species from all these points. We can just see we're slightly looking at the Anaga Peninsula in a slightly different way here. Now, so we sampled 5 individuals per site and generated genomic data to look at patterns of relatedness in a similar way to what I showed you just before. Here is the representation of individual ancestry. So we see a lot of individuals that are purely assigned to one or the other of the ancestral populations that correspond with east and west, and a bunch of individuals in the middle here that seem to be mixed. Now look where these individuals are. These ones are here, these ones are here, these ones are here, and these ones are here. So that spans a distance of less than a kilometre. So we're having all of this interaction happening in less than a kilometre. So the question is is this ancestral admixed populations, or is it something that's happening now? Well, here's a really simple representation that actually speaks volumes. Here's a PCA of the variation. So we have, these are the individuals from the western pure. These are the individuals from the the eastern pure. And in here, which spans most of the first principal component, we have all the individuals that are admixed. So this is exactly what you'd expect if hybridization is happening now, and it is exactly what is happening. We have these two populations are now in contact and hybridizing, and we can demonstrate that empirically with simulations where we can simulate what a hybrid should look like from the parentals, and we find that, yes, we're finding first and second generation back crosses from the small samples that we have. And what we see, this is looking at loci that are fixed or close to fixed in either of the parental populations. We see sharp climb of transition across loci, across this contact zone. So the question of are these things species? Yes, they are very, very much behaving as species in terms of reproductive isolation. Further, we modelled the demographic history of this complex using a program called DARDI that we have our empirical, a summary of allele frequencies. I'm not going to go into detail how it works. And we can test different demographic models and see which our empirical data fits best. The simplest scenario is just simple isolation, no secondary contact. The more complex model is isolation, contact, isolation again, contact again. And across all the models, it is this model that is the clearest, the best fit to the data, closely followed by another model which involves just one cycle of isolation and secondary contact, which is quite surprising for me that our data has enough information to actually resolve between these two models. So it's telling us that there's a repeated dynamic going on of isolation and secondary contact. Okay, and it's also telling us that these things are mostly in isolation. There are small periods of contact, but most of the time it's in geographic isolation. So what all the data is telling us is that most of the time the beetle species that are inhabiting this wet cloud forest are in isolation. Most of the geological history, they're in periods of isolation. Then Sometimes they come into contact and then separate again, okay? Now we know that we are in a period of contact now because we have ongoing hybridization within this particular species across this region of interest. And this is where we are now in time. We are in the warm period, in interglacial. So this is telling us a lot. It's like a really nice, really nice data on what we can on what the climatic consequences are for the cloud forest. The only explanation is that in cooler time periods, the cloud layer is, in cooler time periods, the cloud layer is much higher and creating isolation by drying out these lower elevation areas of the distribution. And this fits quite well with, I mean, there are competing models, but one of the models that came out some years ago was predicting that as the climate warms, the cloud layer is likely to move down slope, creating more humid forest. In cooler interglacial periods, the cloud layer should move up slope, creating a geographically less expansive forest. And this phenomenon has having repercussions across the entire species community that we're looking at within these laurel forest beetles. Okay, I think there's a lot more implications to that, but I'm just going to have to draw it to a close there. And these are our collaborators that I should also acknowledge who've contributed in some way to this research. Okay, thank you.
Well, thank you, Brent. There is time for a quick question, please. No questions? Oh, yeah. Richard?
Thank you, Brent. I would like to. Hi, Brent here. Hi. The landslide you were talking about, I suppose the Taganana landslide or similar. When was it?
It's estimated between 4.1 and estimated to be between 4.1 and 4.7 million years ago.
Okay, then it agrees with the oldest dating divergences of the oldest phylogroups of beetles.
Yeah, there's that one species that matches with that, all the rest post-date the event itself. I think when you look at the data, most of the species show signs for a more younger onset of divergence. That's probably because closer towards the present through the Quaternary, climate oscillations became higher, so the exaggeration of the separation of those two east and western extremes would have been more severe. So it's quite possibly telling us something about the ecology of the species. That let me think about that. The more species more reliant on a humid environment were probably being impacted earlier on in the cycle. Yeah.
Thank you.
Thank you again. We don't have more time for questions to not be too delayed with the schedule. But I would like to thank again Brent, please. Our next speaker is Zach Carter.
I'm going to move the microphone over to stick to a script, otherwise I'll be here all day. So. Okay. Can you guys hear me all right? Okay. So thank you for having me. I'm excited to be here to present my research. It's part of my PhD project. I conducted this research alongside James Russell, who is here somewhere, and others, and George Perry at the University of Auckland. So this study we are titling Determining the Underlying Structure of Insular Isolation Measures. So geographic isolation is generally measured for a particular focal island with respect to a nearby source location under the context of a dispersing organism, such as a rat. So a source location is one that harbors a substantial population of potential migrants, and it could be something like a mainland, such as what that's pictured. So in this sense, an island's level of isolation is equivalent to its accessibility from that mainland source location. Where an isolated island is one that is also inaccessible to a particular organism. So, oftentimes a practitioner will measure isolation simply as the straight line or Euclidean distance that separates the source location and the focal island. So comparisons of the difference in magnitude can then be made between two or more locations. However, we can intuitively discern that this is likely a misrepresentation of the dispersal trajectory, since most organisms do not necessarily travel as the crow flies, unless you have a flying rat. So moreover, a straight line distance measurement would consider two islands that are equidistant from a source location to be equally isolated, even if one is located in a dense archipelago and the other is standalone. So in this latter case, we can almost guarantee that an organism's dispersal path will not consist of a straight line. So this invariably changes the island's level of isolation, and this may need to be reflected in its actual characterization. So to accommodate the nuances associated with variable dispersal trajectories and differing network orientations, there's been a proliferation of metrics describing insular isolation. So this largely started in the mid-1960s with MacArthur and Wilson's seminal work describing the drivers of insular species richness on islands. So the sheer variety of measures available have provided practitioners with a wealth of information, but this also has created a potential source of confusion. It can be difficult to discern how many components of insular isolation are relevant and which variables should be used to represent those components. So if we were to reliably characterize isolation, it is critical that these measures be described parsimoniously. And so that's what we sought to do with this research. So the goals of our study were twofold. First, we sought to determine the underlying structure of multiple measures of insular isolation. In other words, we wanted to determine the different components of a robust measurement. And of those components, we wanted to find measures that suitably represent them as a way of facilitating measurement for future studies. And second, we wanted to test the discriminative power of these components using an insular data set. So we conducted the study in the New Zealand archipelago. New Zealand has 4 main source land masses, including the North Island, as well as the aptly named South Island. There's also Great Barrier and also Stewart Island in the south. Interestingly enough, the majority of New Zealand's islands, offshore islands, are located in 4 main groups as well, including the Hauraki Gulf, Marlborough Sounds, Fiordland, and Stewart Island. In total, there's 890 offshore islands in New Zealand that are 1 hectare in size or larger. We did not include outlying islands in that those were separated from offshore and that they are 50 kilometers or greater from the New Zealand mainland and are also not connected to the continental shelf. And so this provides a slightly better picture of all the different islands. So you can see here this is Great Barrier, which we are considering as a mainland, as well as Stewart Island down here. And so multiple of our measures were context-specific, requiring behavioral details of a particular dispersing organism. We selected species that are subject to New Zealand's predator-free 2050 program, which includes rats, including Rattus rattus, Norvegicus, and Excellans, mustelids, including Furo, Erminia, and Nivalis, and also the common brush-tailed possum. So combined, these species are the country's most harmful pests and are being targeted for complete eradication by 2050. New Zealand biota evolved in the absence of mammals, so any mammal that is actually present and predates upon the organisms have a significant impact. So they've been eradicated from roughly a third of offshore islands. However, they are capable reinvaders due to the propensity for overland and water dispersal. So understanding an island's level of isolation provides insight into the potential for reinvasion, which is critical when prioritizing eradication projects and for determining suitable biosecurity measures. So in this case, highly isolated islands are likely to be good eradication candidates, and they are less accessible than those that are closer or more accessible from the mainland. So this was the frame of reference for our second objective, and I'll explain that further in depth in a few slides. So this slide has a lot of moving parts, so hopefully I'll be able to explain it suitably. So all the measures that we conducted, that we investigated, were conducted using Python, GIS, or in R. The first method is a simple Euclidean distance, which was measured as the shortest distance between a focal island and the mainland. And next, we investigated the least cost approach, which influences dispersal trajectory via landscape connectivity. So connectivity is defined as the degree to which a landscape facilitates or impedes movement. These factors can include structural connectivity, that are landscape characteristics, and functional connectivity, which are the organism's specific biological characteristics. So the least cost approach assumes the organism has perfect knowledge of the surroundings, and as such, the organism will make the most efficient path possible from source to destination. And to represent this, we used a cost surface raster for the movement process. Values in the cost surface represent the per unit cost of dispersing through the landscape, and were based upon selected functional and structural factors. We decided to choose very broad factors that minimized traversal over water, as well as vertical cliff faces, so that our methods could be used in other studies in the future. Next, okay, we looked at the stepping stone gap, and stepping stones are a common biogeographic feature where they decrease isolation by increasing biotic exchange. So we wanted to look at the maximum over water distance that an organism has to travel, assuming that is what the organism will not want to do while it's emigrating. And then next we looked at the distance to the nearest life-supporting island. We considered that to be any island greater than or equal to 5 hectares in size. It's important to note that the island does not have to be the mainland. It could be any surrounding island that is suitable for this characteristic. And so similarly, we also looked at the average distance to the nearest 5 life-supporting land masses. And again, those are anything greater than or equal to 5 hectares in size and do not have to be mainland New Zealand. So for the non-distance metrics, we looked at island area. An island area is not necessarily a measure of isolation per se, but area is not independent of an island's isolation. I'm sure we're all familiar with the target area effect, where as an island's size increases, its isolation tends to decrease. So we thought including this would be a good idea as well. Next, we looked at landscape isolation buffers. We used 3 of them based on dispersal characteristics of the organisms that we were looking at. So we used 1 kilometer, 3 kilometer, and 5 kilometers. And this takes into account the amount of land surrounding the perimeter of the island of interest. And this is supposed to facilitate understanding the island's isolation in a highly congested archipelago. And so then next we looked at the stepping stone count, so the number of stepping stones associated with dispersal trajectory. So you can see here, it's kind of hard to see, they're highlighted in yellow. So for this particular path, it used 2 stepping stones, 1 and the second one here. And for this particular dispersal trajectory, it only used 1 stepping stone. And then next we looked at the accumulated cost, which is associated with the least cost path model. And both of those were affected by a digital elevation model and an ocean current model, which I talked about previously. And then finally, we looked at commute distance. And so this is antithetic to the least cost assumption that an organism will use an optimal dispersal pathway. So these models are based on electrical circuit theory, where the landscape is thought to be a web of resistors connected by edges. So each resistor represents a local dispersal resistance, and localities that are highly conductive or have low resistance will therefore most facilitate movement. So as an organism disperses from a source location, travel will then be focused through those conductive areas. And so you can see that here. So I tried to show a picture, and so you can see that the organism will most likely travel through areas that are not resistant. So this could be, for example, a cliff face where the organism will try to go around because it's the easiest option. And so we used a PCA to reduce the dimensionality of our variables. So based on the variance of the data, the PCA gives new latent variables characterizing the primary components of isolation. I'm sure most everyone is familiar with a PCA, but essentially what it does is it looks at the variance of the data set. So if you plot variable 1 against variable 2, it will investigate the amount of variance. So it is the most variance here is across this axis, where the second is tangential, is across this axis. So then the axes of the PCA will become these axes identified. So you can see this is rotated. And so we also conducted a MANOVA to assess the discriminatory power of our isolation measures, a simple MANOVA. So we detected for differences between islands that are rat invaded and those that are rat eradicated. We chose to focus this part of the analysis on rat species because they're New Zealand's most widespread mammal and because the residency on islands are more permanent than other species that have greater mobility, such as stoats. So we also restricted the analysis to islands that are greater than or equal to 5 hectares, where presence data was reliably available. So in total, 67 offshore islands were rat invaded and 74 have been confirmed. 67 islands have been rat invaded and 74 have been confirmed eradicated. So identifying the dichotomy in these groups simultaneously assesses the discriminatory power of our measures and validates their applicability as an eradication prioritization tool. So this is essentially the results of our study. So these are the composite measures of isolation. So you can see that we only have 3 PCs listed. The 4th PC comprised had an eigenvalue less than 1, which means that the measures, or the component explained as much variance as the measures being input. So essentially they were superfluous. So variables that clustered on the same PC, you can see here, are mostly distance-based measures. So we entitled this distance. Second was stepping stones. These 2 clustered the most. And then 3, we call this insular network, which is an interesting, it shows a dichotomy between area as well as, it's an endogenous and exogenous, where it shows an internal feature of the island area, as well as the distance to the nearest islands surrounding. So note that the results for PC1, that any of these, including a simple Euclidean distance measurement, is likely sufficient when quantifying island distance offshore. The connectivity-based, least cost path, and circuit theory metrics do not explain more data set variance in this case. So I'll touch more on this soon, but overall the high degree of redundancy in the loaded variables suggests that single variable representatives can be used to characterize each identified component. So for the discriminatory power of our isolation measures, there was a significant main effect of isolation on groupings of rat-invaded and rat-eradicated islands. You can see that here in the biplot of the PCA, you can see that the rat-invaded islands are much less isolated when compared to islands that have been rat-eradicated. So this outcome demonstrates the discriminatory power of our isolation measures in that our characterization can successfully differentiate between actually occurring groups. I'll draw your attention to the island down here. This is White Island in the Bay of Plenty, and we'll talk about that more in a second. So this slide is full. So I'll talk about the interpretation of the clustered variables and why a straight line distance measurement may be actually equivalent to more complex methods. So although circuit theory and least cost methods are effective tools for modeling connectivity, they apparently have fewer advantages for measuring the isolation of islands. So there's likely 2 reasons for this. And first, it's the effect of structural and functional connectivity are apparent only if the landscape being considered is heterogeneous. In our case, the seascape surrounding each focal island was often homogeneously inhospitable. So this homogeneity meant that our distance-based connectivity metrics tended to approximate a straight line path in that it was the most efficient option available. Because the least cost path method assumes that the animal has a complete understanding of its surroundings, it will take the path of least resistance. And so second, for connectivity metrics, including the accumulated cost and commute distance measures, they are directly proportional to the Euclidean distance when they're in a homogeneous environment because the weighted edges of the cost surface raster are uniform across the environment. So I tried to show that down here, where here it's a heterogeneous environment and here it's homogeneous. So in a typical heterogeneous or terrestrial environment, each weighted edge is calculated as the product of the average cost between the connected cells and the Euclidean distance that separates them. And so each weighted edge is then summed if it is used in the traversal path. When each connected cell is weighted equally, though, the total accumulated edge summation simply becomes the product of the uniform edge weight and the Euclidean distance between the source and the destination. So overall, our connectivity metrics could not explain structural or functional traits in a manner that provided additional dimensionality to the existing characterization. So as such, they became highly redundant with other distance-based measures and ultimately clustered on the first principal component. So for interpretation of PC2, so although PC2 was calculated using connectivity metrics, we'd like to note that the stepping stones used to access the focal island were often readily identifiable. So this suggests that a practitioner wishing to calculate this measure can do so without the use of biologically informed assumptions. Also, the positive loading scores on PC2, which you may have seen when we showed the results, suggest that the measures are seemingly confounding because they have positive correlation. However, overall, they reflect the number of stepping stones intersecting the least cost path, where quantity is directly proportional to offshore distance. So you can see that as distance decreases, the number of stepping stones starts to funnel. And for PC3, the interpretation, recent work has substantiated that inter-archipelago configuration strongly influences the dynamics of vertebrate press dispersal. So or just vertebrate dispersal in general. So PC3 insular network is consistent with this finding, demonstrating that characteristics beyond mainland accessibility are important when considering island isolation. So although the characterizing variables of PC3 are seemingly opposite in their representation of endogenous and exogenous insular characteristics, either can be used to better understand this configuration. And I'll briefly talk about application across taxa in landscapes. I don't think I have too much time left, so I'll go through this quickly. Essentially, what we think is that these methods can be applied to different taxa as long as the dispersal scale, as long as what's changing is the dispersal scale. So in this highly unscientific figure, you can see that we're going to smaller dispersal scale, where we have the New Zealand weta, to the larger scale of the New Zealand sheep. So, but as soon as the dispersal mode changes, where you get something like a flight, or there's a Kia, these measures likely will not be applicable. And I think this would be an interesting study, an interesting follow-up study to see how they actually compare. So across landscapes, unlike some other studies, we did not find support for the landscape buffer as an adequate descriptor, and this is likely due to the factor characterization of our PCA. So multiple other authors have found that support for this measure and presume that it would more accurately reflect isolation in a congested archipelago. So we decided to rerun the PCA for islands only in the Fiordland region, which is New Zealand's densest archipelago. So this was 141 different islands. And indeed, the 1 kilometer buffer explained the most variance for the second principal component, but Euclidean distance remained as the first. And so implications of the component discriminatory power, so based upon the PC1 and PC2 biplot coordinates, Radix excellence is counterintuitively present on the highly isolated White Island. So I show this somewhat differently here, and you can see White Island here is an outlier within our group. So in the midst of New Zealand's aggressive predator-free 2050 timeline, this appears to be an anomaly. So our metrics have instead likely identified that mainland accessibility is not what is limiting eradication success on this island. Other factors must be at play. So, and indeed, White Island is actually an active volcano and is privately owned, and it is not actually a conservation priority by the government. So discerning reinvasion potential is an important aspect of every eradication program, and our isolation measures can be used to help understand this concept tangibly. So for the take-home messages, in the context of terrestrial mammal dispersal, we found the underlying structure of insular isolation measures with 3 distinct components, including distance, stepping stones, and insular network. These components can likely be represented using a single variable characterization. We recommend Euclidean distance from the mainland, the number of stepping stones along the dispersal path, and island area, because the variables are not biologically informed, meaning they can be applied without extensive functional and structural assumptions. And finally, our components have application to assist conservation efforts, and that they can identify plausible eradication candidates that are unlikely to be reinvaded, thereby maximizing input. So thank you, and I'd like to thank my supervisors, James Russell and George Perry. Be happy to take any questions.
Thank you, Zach. There is time for, well, there is no time, but if someone has a very quick question, please go ahead. No quick question? Yeah. Let's thank again, Zach, please. Our next speaker is Tania D'haijère, and she's coming from University of Brussels.
Hi everybody, thank you for having me here today. So we're going to São Tomé and Príncipe. So São Tomé and Príncipe are part of the Cameroonian volcanic line in West Central Africa. This line goes from Lake Chad up to Annobon, and in the archipelago there is one continental island, Bioko, and then 3 oceanic islands, Príncipe, São Tomé, and Annobon. So Príncipe is the oldest of the oceanic island. It's 31 million years old, and it's the closest to the continent, as it lies 216 kilometers from Equatorial Guinea. And it's a biosphere reserve of UNESCO since 2012, the whole island. São Tomé is younger, so 13 million years old approximately, and it's a little bit further, as it lies 239 kilometers from Gabon. São Tomé and Príncipe are mainly known for their birds, as it is the archipelago with the most number of birds per land area. So there is 17 species endemic to São Tomé, 8 to Príncipe, and 3 shared by the 2 islands. And in comparison, in Galapagos, it's 22 species of birds for a land area 10 times greater. So it's quite impressive. And here you have the smallest ibis on Earth, and here the biggest canary on Earth. Among plants, there is still a lot to do. There is not a lot known. So there is an endemism rates of 14% for both islands. And the 3 main families in terms of number of species and endemic species are the Orchidaceae, the Rubiaceae, and the Euphorbiaceae. And the Polystachya and the Psychotria are the 2 main genus with the most endemicity. And among plants, you have study about begonias, because there is 6 species of begonias endemic to São Tomé and Príncipe, and also impatiens, where there is 3 species on 3 that are endemics. So my research are focused on Tridactyle, so it's a genus of orchids. It's approximately 50 species. It's all sub-Saharan genus, and it's found in the archipelago, the continental Africa, but not Madagascar. Among these species, 10% are found in São Tomé, and 50% of them are endemics. So that leads to our question, so to understand what scenario brings this endemicity, this high endemicity there. So we wanted to do phylogeny, to have the phylogeny of the Tridactyle, to compare the morphology-based species to the clade obtained in the phylogeny, and then compare with the result obtained for other plants. So about our sampling methods, we have developed for 20 years now a shade house network in the whole Africa, so also in Madagascar, that allow us to have 30,000 of plants that have been cultivated since 20 years, and 24,000 specimens for herbarium with silica gel, pictures, and so on. And that allows us to go into the forest to kind of rescue the orchids from fallen trees. And so we usually found them sterile and without flower, and then we grow them in the shade house to collect the flower and identify them better. And so if I really need one species, then sometimes we go into the tree with somebody that really knows how to climb, or myself, and so we take like one individuals out of 5 or 10 to not over collect among populations. So this allows us to have 200 samples of Tridactyle from 40 localities in Central Africa. So the method is genome skimming, so low coverage of the whole genome and then focusing on chloroplast and ribosomal. And unfortunately for me, our coverage was a little bit too low, so we had to remove quite a lot of bases, but for the first run I did, and so we ended up with 25,000 base pairs for the chloroplast and 3,500 base pairs for the ribosome, and we analyze it with star beast. So in total we have 1,600 SNPs. And this is the phylogeny I obtained. So you have the outgroups, in orange the Orchidaceae, in purple the Epidendroideae, and then inside of the Epidendroideae, the Vendae. So here you have these two species that we have recombined in another genus, and it's an article that is in press now. And so we dated this phylogeny using the Givnish datation. So Givnish did the whole datation of the whole Orchidaceae family, and he did quite a good job, so we used that. And so this is the real Tridactyle. And here you can see that the morphous groups, that are the boxes of color, match pretty well with the molecular data. So by example, here you have the long inflorescence in blue. In light blue, you have the long inflorescence plants, different species, but still with a morphological character common. And here you have all the species of the archipelago. So they form one clade here, with species in orange found on the continent, species found on São Tomé in dark blue, light blue on Príncipe, and then in green found in the continent and the archipelago. So so here you have the morphogroups and where you found the plants. So there is some that we found in Central Africa, here too, West and East Africa. So about our scenario, the first one we thought of was radiation, but then we would have all the species of the continent at the base, and this is not the case here, so that's probably not the real story. The other thing we thought was the diversification of different groups, but this is not very parsimonious, and then it raised the question why in this particular clade of the tree and not the other one that are also in Central Africa. So the hypothesis that we thought is probably the best explaining the data we have here is two events of colonization of the island, and then maybe going back on the continent at some point with diversification or not. So here it's not well resolved, so it's not really clear, but that's what we thought is the most probable for now. So also about ecological consideration, among these clades, so it's this one and this one, you have one species on very open area, almost not forest, even really close to the sea, and then the other one at the really higher altitudes, rainforest, rain cloud forest. So probably there is an effect of the habitat that drives speciation, maybe, that's a possibility. And about the other plants that have been looked at in São Tomé, so there is multiple events that have been proved and that are pre-Pleistocene. So it's one event for green rhododendron because there is only one species. So we can't know much about it, but so it's pre-Pleistocene. It's from approximately 7 million years old. And yeah, the multiple events here for Begoniaceae, they are still grouped in one clade, kind of what I've got. So yeah, that's it for the plants. And about animals, so there was also one or multiple events, but much more recent, so really Pleistocene, much more like what I obtained. For the Drosophila, the history is that there is different endemic species among the Cameroonian volcanic line on the continent mountains and in the islands. And they found out that the endemic species derived from the continental species, and that there was a very recent recolonization from the continental species to São Tomé. And that's also the story for the populations level of the gray parrot. And that could also be a possibility for the Tridactyle latifolia and the species C. So basically, I have still some work. It's an ongoing project. So I had the second run of NGS that we've done in June. So I have now the complete data set with more West and East African orchids and also South African one. And I will use diverse methods to assess the center of diversification and how, well, the real scenario among these species. And also doing a population study for Tridactyles, which is the species with the diversification here with all the one on the continent. So, come on. Yep. So yeah, for the archipelago, so it has probably acted as a refuge during the during the Miocene, because at least for the begonias and the green rhododendron, there is this evidence that it was probably more paleoendemism and relicts from lineage that were on the continent. From the 10% of the genus, it's actually 20% of the genus that are found on this island, with 75% of endemicity, which is way much bigger than we thought. So here we have a real bias on the taxonomy that is not well known, and not just in São Tomé, everywhere on the planet. And then there is a good side in São Tomé and Príncipe that 30% of the two islands is a protected area. It's the National Park Obo, so at least my orchids are quite safe compared to other places on the planet. And I want to thank everybody that helped to raise this project. And I thank you.
Well, thank you, Tania. We have time for questions.
Hello. With the lineage that you think that are paleoendemicians, they've been extincted in the continent. Is there evidence of continental extinctions or not?
Okay, for the other lineage, like begonias,
Begonia.
Yes. For the Tridactyle, because we really extensively sampled Central Africa. If we don't find it on the continent, yeah, that if it's paleoendemism, definitely it's extinction on the continent.
Thank you.
You mentioned, Tania, that the archipelago, and in particular, I think Puerto Príncipe, São Tomé, they are amazingly rich in endemisms compared to Galapagos. So what is your hypothesis? Why it's so rich in birds and other organism groups? Is only because of these refugia during the pre-Pleistocene and Pleistocene, or because it's closer to the continent? What would be your hypothesis to explain that?
Yeah, so basically the fact that it is so close to the continent compared to other archipelago really plays a role. And yeah, so this and the fact that São Tomé, even with the volcanism, is quite big, like 1,000 kilometers square. So even with volcanism, the climate didn't change that much there. So the volcanism obviously act for bottlenecks in species, but still it was kind of safer place. Príncipe, totally not. It was really not. It was really disturbed until recently. So, but for São Tomé, yeah, I think that act as well.
More questions? Well, actually, I have another one. The species of your group, do they disperse by wind?
Yes, so all orchids have the tiniest seeds in the angiosperm group. So yeah, they disperse by winds for the seeds. And for pollination, we don't know, probably small
That was
moths.
my second question.
Okay.
So I was assuming that they disperse by wind, and I wanted to know, following up
So
with the talk of
pollination-
Susan.
Yeah, so for pollination, I only seen one moth on it and one tipulid, but there is a big missing bibliography on the pollination of these small spurred orchids compared to the big one that everybody knows it's related to the big moth and so on. Here it's really still a gray area.
Any other question? Okay, thank you, Tania. So our last speaker of today is Alain Vanderpoorten from Liège University. And I didn't say that in the presentation, but if there is time left, then we are going to have the opportunity to bring all the speakers to the stage again. And if you had some pending question, there will be opportunity to ask.
So, hello everybody. Thanks a lot very much indeed for staying to this last talk. Thanks a lot also, as I said before, to Dominique, Olivier and Claudine for accepting our symposium on dispersal on islands. And I must confess that my talk is a bit more about dispersal than it is about islands. But here we go. So the topic of today is how we can predict trend shifts in wind dispersed organisms under changing climate conditions. And so I guess that any talk about climate change has to display at least one graph on the IPCC. So the one that I choose to display today is a graph showing the global increase of temperature on Earth since the pre-industrial era. And this graph shows that since 1960, the Earth has already undergone a warming of, on average, 1 degree. Of course, there is a substantial geographic variation because, for instance, in the Arctic region, the warming has already been of 3 degrees. And this trend is expected to continue in the future with an intensity that depends, of course, on the different climate scenarios that have been proposed. And therefore, climate change has been identified as one of the major biodiversity threats leading possibly to the 6th mass extinction on Earth. And so one of the most useful, or I would say widespread method to infer the impact of climate change on species distribution is by implementing species distribution models. So in two seconds, how this works, you start with sampling points, so data points that document the distribution of the species in the landscape. Then you combine this information with environmental layers, climatic data, pedological data, whatever. And then you derive for each variable a response curve. Then you combine all these response curves together to provide a multivariate niche that is eventually projected on the landscape, and you can use different climatic layers for that projection. You can use current climatic layers, or you can also use future climatic layers to predict indeed what is the impact of climate change on the species distribution. And indeed, these projections are a good reflection of what you can expect in terms of future distribution of your species, provided that a series of assumptions are respected, including that there are no biotic interactions and also that there are no dispersal limitations. And of course, if your purpose is to determine whether species are able to track suitable climate areas in the future, it's a bit annoying to think that a species distribution model do not take dispersal at all into account. And to show the impact that this can have on our ability to make predictions for the future, I took an example of a study that was published in 2005 about the predicted impact of future climate change on the European angiosperm flora. So here, each bar represents the proportion of species that are predicted to be at no risk in black in 2080, and in the different streak patterns represent the proportion of species that are predicted to be at different, that are expected to experience different threats in the future as defined by IUCN. So these predictions are made for different competing climate scenarios, and they are made for the two major assumptions. On the left, we have a no migration scenario, which means that even though a pixel would become suitable in the future for a species, it would not be colonized because the species cannot move to that pixel. And on the right, we have the reverse scenario, the full migration scenario, according to which as soon as a pixel is suitable in the future, it will be readily colonized by that species because it has no dispersal limitation. And of course, we can then see that we have striking differences between these two assumptions. On the left, we have more than 50% of the species that are predicted to be vulnerable or even committed to extinction by 2080 under the no migration scenario, whereas under the full migration scenario, we expect that about 70% of the species will be at no risk. And of course, this creates a huge uncertainty that weakens our ability to predict what will be the impact of climate change on species distributions. And therefore, there has been an increasing interest to develop integrative models that combine information on ecological niche and also information about the ability of the species to disperse in the landscape. One of these techniques is called MICCLIM, and it's very convenient to talk about MICCLIM, the BioClim auditorium, by the way. So MICCLIM was developed by Englert and Guisan in 2009, and it starts with information about the species distribution. So the blue layer that we have here is just a grid documenting the distribution of the species in the landscape. And then you use this distribution and you combine this information on species distribution with information on climatic conditions, which allows you to predict the distribution of the species in the present, but also in the future. And so you basically produce maps of habitat suitability through time. And then what you want to do is to make simulation of the dispersal of the species in this changing landscape. To do these simulations, MICCLIM is going to use two kinds of information. First of all, a short distance dispersal kernel, and a kernel is just the probability of dispersal depending on the distance from the source. And it's also going to use a long distance dispersal. It's going also to document the long distance dispersal of the species. And so you need to inform MICCLIM with the maximum range that the species can migrate. And also you need to inform it with the probability that these LDD events will take place. So then you perform a series of simulations. I would say that normally you do that at the time scale of 1 year. So every 1 year you perform a simulation of dispersal in this landscape. And eventually after a series of simulations, so you end up your simulations when, for instance, in 2050 or 2080, when you want to estimate the potential distribution of your species in the future. Then you end up with a map like this, where you have in yellow the extinct pixels. You have in pink the suitable cells that remain unoccupied because of dispersal limitations. In black, you have all the species that were and that will remain occupied in the future. And then you have different colors that tell you about the pixels that will become colonized in the future and will remain colonized until the end of these simulations. So our purpose was to apply MICLIM to wind-dispersed organisms, and of course the big problem that we have with wind-dispersed organisms that even though we try to use big binoculars, it's very hard to do direct estimates of dispersal, and therefore you need to use indirect estimates of dispersal. So in our case, we use the wild dispersal model, which was developed by Catulle in 2005 to describe the movement of wind-dispersed seeds. And the wild dispersal model includes two kinds of variables. First of all, you need information about the landscape and the suitability of the landscape for migration. So for instance, of course, the average wind speed in the landscape, and also the canopy height, which is going to restrict the movement of diaspora in the air. And then you need information on the species-specific traits that will modulate the dispersal capacities, and in particular Z0, which is the height of release, and VT, which is the settling velocity. Settling velocity can be easily measured with a fold tower experiment like this, which is associated with a high-speed camera. And we use this experimental design to document the settling velocities of spores. And on the right here, we have the relation between the settling velocity and the diameter of the spore. The black line now is the regression line that we observe from the data points, and the red line is what you would expect in terms of settling velocity under Stokes' law, which describes the movement of a perfect sphere in the air. We can see that there is an almost perfect relationship between settling velocity and diameter, which is very convenient, because then it means that you don't need to go through all this process each time that you want to make some simulations. But, and then I open a small parenthesis here, it's interesting to observe that the biggest spore often depart from the expectations. Some fall down faster than what you expect, and some fall down slower than what you expect. And we are currently investigating what are the mechanisms explaining these differences, and we think that it might be related to spore ultrastructure. So today we try to relate the impact of variation of spore ultrastructure for the dispersal capacity of the species. I close my small parenthesis here, and so the idea now is to implement this wild dispersal model into MICLIM to predict the impact of climate change in wind-dispersed organisms. So this is a spatially explicit model. So if you study a landscape like this, like Europe, you're going to divide it into pixels. For each pixels, you are going to record the information about the extrinsic pixel-specific condition that will affect dispersal. So for instance, wind speed and canopy height. And then you're also going to bring information on species-specific traits that affect dispersal. So the height of release and the settling velocity. Then all this is combined into the wild dispersal model, and then you end up with spatially explicit kernel maps that document the probabilities of dispersal in the landscape. So therefore, we developed MICLIM 1.7 as a spatially explicit model of range shift as a function of climate change. So once again, we start with data points that are used to build species distribution models. These species distribution models are then projected onto extant and future climatic layers. And then, of course, a data point that ends up in a pixel that is unsuitable for the species in the future is a local extinction rate. And then we also use the data points to as starting points for a simulation of migration in this changing landscape. And so any dispersal event that ends up in a suitable pixel is a successful colonization event. That will be the beginning of a new dispersal event at the next simulation. And all in all, what you do at the end of all the simulations is just counting the number of local extinction and local colonization events. So let me show you now an example of application in the case of the European bryophyte flora. So what we did was to sample representative species for the four main biographic elements, so Arctico-alpine, Atlantic white temperate, and Mediterranean. For all these species, we model a species distribution from climatic predictors using ensemble models, using GLM and GBM techniques. And then we projected all these models onto future climatic layers. So let me show you an example of a Mediterranean species like this. We project the model onto future climatic layers. So here we have the future distribution under two climatic change scenarios. And then we partition this potential distribution in the future into three areas. So we have here in green the area that is conserved through time for the species. We have here in blue the area that is expected to be gained, so newly suitable areas in the future. And in orange, we have all the lost areas. So now we did this, of course, for all the biographic elements and all the species. And here we have the proportion of pixels on average that are expected to become suitable in the future, and the percentage on average of pixels that are expected to be lost for that species in the future. So if we look now at Mediterranean species, we see that we have an average increase of suitability of 40% as compared to an average decrease of suitability of about 24%, so a net predicted increase of potential distribution in the Mediterranean, whereas for the alpine Arctic species, we have an increase of suitability of 14%, but a decrease of 39%, so a net decrease of expected suitable areas in the future. I open a second small parenthesis to comment on this, because for those who are familiar with European biogeography, this is a bit unexpected, because that's a map showing the local extinction rates in European angiosperms from the paper that I showed before, the 2005 paper, and we can see that the highest extinction rates are predicted for the Mediterranean. So why the hell do we see a highly threatened pattern for angiosperms, whereas we see the reverse for bryophytes? And the answer to this is probably that 60% of the Mediterranean endemic angiosperm species are very narrow endemics, some endemics to very small areas, and whereas in bryophytes, the distribution range is across the entire Mediterranean area. And because we build the species model, distribution model on basically the climatic suitability that you observe at each data point, of course you infer a much wider niche for a widespread species than you infer for a very endemic species. So we now use these maps of suitability through time to perform our migration simulations. So now I'm just going to show you the migration wave that we observe for a given dispersal kernel at 10-year intervals in Europe, so 2020, 2030, 2040, and 2050. And on the right now, we have the distribution of the pixels that are suitable, but which are uncolonized at the end of the simulation. So the pixels for which the climate is suitable, but which are not expected to be colonized because of dispersal limitations. Let's look at that now into more details. We have now, for each of the biogeographic elements, the rate of colonization as a function of the long distance dispersal probability, 0, 10-4, 10-2, and also the function of the rate of release and the wind speed that we implemented. So average mean speed across the past 30 years and average maximum wind speed across the past 30 years. What is interesting to see here is that one of the most important factors that drives the colonization success is the rate of release. So the colonization success is about 2 to 10 times higher for a release rate of 1 meter as compared to 3 centimeters. And this suggests that the release rate impacts a lot on the colonization success, suggesting that there is a strong evolutionary pressure to elevate the capsule above ground. Most interestingly also, you could observe that the rate of colonization, maximum rate of colonization that we observe with all these dispersal kernels is only 1%, which is really not a lot. And to completely shift the order of magnitude of the colonization event, you need to set the long distance dispersal probability at 10-1. Then just to show you visually how it looks like, that's the end of the simulation in the Mediterranean element when we set the LDD probability to 0, and then we end up with 817 successful colonization events. And here that's the same, but when we set the LDD probability to 10-1, and we can see that even indeed there are striking differences in terms of the colonization ability depending on the LDD capacity, long distance dispersal capacity probability that we set up. I can hear people from the front row saying, don't worry, Alain, anyway, bryophytes have the dispersal capacities to overcome this problem and to track areas of suitable climate. And so they are not dispersal limited, so they won't be concerned by the ongoing climate change. Let's look at this into more detail. So this is now a graph representing the density of spore per square meter, depending on the distance from the source. And this is one of the rare experiments documenting this based on spore trapping experiments. And what we can see is that there is a striking decrease in the spore density until during the first 100 meters. And then beyond that scale, we still have quite a large density of spores, but then the relationship is completely distance independent, as if any spore that can go beyond 100 meters, once it's airborne, it can travel basically across 1,000 meters or 10,000 kilometers. Then on the right here, we have the accumulation probability of spore deposition as a function of the distance for the source for different wind speeds. And once again, we see that we have a huge proportion of spores that are expected to be deposited at long distances. And so, of course, if this is true, then it means that indeed bryophytes are not really severely threatened by climate change because they can really travel a lot. And so this is what we wanted to test now using genetic evidence. So indeed, when you have such fat-tailed dispersal kernels, so when you have such a high probability that spores travel across long distances, then you expect that an inverse isolation effect is going to take place. What is this? It just means that within the source colony, you expect that the bulk of the diaspores will be recruited locally and creating this strong isolation by distance pattern. But as soon as you move away from these source colonies, of course, you get diaspora from many different source colonies, and therefore the genetic diversity per individual will increase with increasing distance, and this is going to completely erase any signal of IBD in your data. This is what Vekmans and Hardy simulated. So here we have the genetic similarity, the kinship coefficient between pairs of individuals, depending on the geographic distance for different values of the long distance probability. And we can see that indeed at LDD probability of 10-1, any signal of LDD is completely erased from the data. So that's, as I said, what we tested with a meta-analysis of studies that documented the spatial genetic structure of bryophytes at the landscape scale. And so what we did to test the inverse isolation hypothesis was to recalculate the slope of the IBD relationships at different distance intervals. So we calculate the slope for pairs of individuals that are separated by less than 100 meters, between 100 meters and 1 kilometer, and so on. And on the left is just one example of a spatial autocorrelagram showing the kinship coefficient as a function of the distance. So what we can see is that at very short distance, almost all the tests are highly significant. We have very steep slopes, which is very consistent with the idea that indeed spore density decreased very quickly during the first 100 meters. But then we can see then the slope is completely flat after that distance, and indeed none of the tests were significant, which is consistent with the inverse isolation hypothesis. But unlike what this hypothesis suggests, as soon as you go beyond 10 kilometers, 100 kilometers, then we cover a significant IBD signal in a high proportion of the data set, which suggests that as soon as you are beyond 10 or 100 kilometers, once again, IBD is important to explain species distributions, and therefore you cannot take the inverse isolation hypothesis as a rule, even in highly dispersive organisms like bryophytes. So even apparently highly dispersive organisms like bryophytes do experience dispersal limitations. I will be very quick here, since we are running out of time. So now the idea is, of course, we know that they are dispersal limited. Can we estimate this probability of long-distance dispersal? That seems to be crucial to explain the ability or not to track areas of suitable climate in the future. So in 2 words, we did a study estimating the post-glacial migration rates of bryophytes between southern and northern Europe. So basically we use advantage of the genetic structure of the data with an ABC approach to infer demographic parameters of interest, and here in particular the posterior probability distribution of the long-distance migration rate, which is the proportion of individuals that will migrate per generation. And as we can see here, the mode of the LDD probability that we documented with this study was about 10⁻², which is about 10 times lower than the rate that you would require to balance extinction and migration rates. All this to conclude that our simulation of spore dispersal on our ongoing climate change point to substantially higher extinction rates than colonization rates. Even apparently extremely efficient dispersals like bryophytes are severely threatened by short-term effects of climate change. And I'm happy to say that indeed in this case, bryophytes are the canaries in the coal mine of climate change. Many people help to produce this study. I'm not sure about the kinship coefficient among these collaborators, but there is a significant geographic structure in the data. So we have Florian Zanata and Flavien Collard, who are PhD students working on the effect of climate change on bryophytes. Then we have Romain Engler at the University of Lausanne, who developed MICLIM, Olivier Brönnimann and Antoine Guisan, who help a lot with the modeling. And then we also recruited some collaborators after some long-distance dispersal events. Thanks a lot for your attention.
Well, maybe there is time for some question. Yes, Paulo.
Thank you for your fantastic presentation. In these models, it's possible to include scenarios of land use changes or changes in the agriculture intensity, forestry changes, to see what will happen with species with specific ecologies.
Sorry?
To see what will be the impact of these scenarios of land use change in specific species with specific ecology.
Yeah, I mean, that's the step beyond. I mean, for the moment, everything is based purely on climatic change scenarios. You're absolutely right. I mean, the next step is also to include more environmental layers, including, of course, land use, disturbance, and so on. Absolutely. I hope we can do that in the very near future.
Well, I see that it's time for coffee already, so I just would like to thank again our fantastic five speakers who are going to be at the 10 or around, so you are very welcome to ask questions if there was something that got behind. So thank you again, and to you for coming.